and the topic is more and more neurons. I think you all know that there's been an enormous amount of excitement about our ability to look at larger and larger populations of neurons simultaneously. And to realize the promise of those things, you need new experiments, you need new analysis techniques, and you need new theory. And so we're going to have 3 talks that more or less hit those 3 things, give you some slice through them. And it's a great pleasure to start by introducing my colleague, Leifer, who's going to talk to us about the elegans.
Thank you, Bill. It's a treat to be here. I've really enjoyed this series over the years, and thank you all. I see a number of friendly faces. Thank you all who took the train, and including members of my lab. So I'm going to be filling in on the kind of C. elegans experimental side of the 3 themes. I'm going to move this out of the way, maybe. See how that works. And today I'll be talking a little bit about how we're starting to think about comparing a nervous system structure to its function. I work in the nematode C. elegans, which you can see here. And I'll talk just a little bit about the worm and why it's a great system. And then I'll be focusing particularly on one project that we're all really excited about in the lab to measure how signals propagate through the worm very systematically. And so this image on the right here, this is these are a collection of neurons in the head of the worm. You know, one of the reasons as biophysicists we're really excited to work in C. elegans is because it strikes this nice balance between simplicity and complexity. So the worm can do things that are very evocative of higher order organisms. So it can navigate a sensory landscape, it can learn to associate odor with food, it can evade predators, it can seek out or avoid potential mates, depending on its mood. And yet it's this nice compact system. So it's only 1 millimeter long by 80 microns wide. It has 302 neurons. We have a lot of confidence in that number. It has less than 1,000 cells total across the whole organism. And it's this wonderful model system that began as a genetic model system. And so we have a lot of fantastic tools to work with it. And you'll see, coming from an optics background, it's optically transparent. So many of our investigations in the nervous system can be converted into a problem of getting light into and out of the worm as we need. So our lab, is interested in a number of questions about brain and behavior, inspired actually in part by Carsen Stringer and others' work, who we'll hear from today. We're very interested in how large-scale patterns of neural activity relate to locomotion. And so our group has done a lot of work on that. We're also interested in how the worm processes information to generate actions. And so we have kind of another thread in the lab that looks at specific behaviors, like how this animal responds to a touch and how the nervous system mediates that response and how that response might change depending on what the animal is doing. And for both of these types of investigations, and for many of the other investigations we have in the lab, one of the things we found is, as we can really characterize the neural dynamics, the animal's movements, or the kind of input-output relationship between sensory inputs and motor outputs, what we're always looking for is some underlying mechanism, something that describes the equations that govern these dynamics, or some circuit level mechanism. And in each case, it would be very convenient if we knew how signals flowed through the nervous system, because presumably it's those signals in the brain that are dictating the dynamics or dictating this input-output relationship. And so that has led us to the project that I'm going to talk about today, which is how do signals propagate through the brain. And so for this project, we're trying to tackle that head on through direct measurement. And before I forget, well, okay, I'm going to go out of order. Before I forget, these are the 3 members of my lab who've been most involved in this project, and they're actually sitting there in the back row. And I'm showing their glamour shot because we just had the Neuroscience Institute just hired professional photographers to take photos of everybody. Oops. And so there's their glamour shot. So this is the work done by Dr. Francesca Randi, Sophie Diwali, and Dr. Anu Sharma. And they can help correct any questions because they're right there. Okay, so the goal of this project is to measure signal propagation at brain scale through the nervous system with an eye to do this in a very comprehensive and systematic way. And the goal is really to provide a resource for understanding the circuits we're interested in, or for modeling, which, for adding additional constraints to try to understand network dynamics. So that's one goal. And then the other goal is to leverage the fact that in the worm, we're in this unique situation where we have both the connectome and now the ability to make these functional measurements. So we're uniquely positioned. We have this privileged position to compare structure and function in a way that's you can't do in many other systems. And then I'll talk a little bit about one of our more salient findings, which is that extrasynaptic signaling that doesn't appear in the connectome seems to play an important role in dynamics. Okay, there's our amazing team again. And I think I think we have to stare at it for 10 seconds because I put in some thing where it has like a 10-second, there we go. Okay, good. So let's just start with the C. elegans wiring diagram because C. elegans was the first organism, and I would still argue is the organism with the most comprehensive wiring diagram or connectome. And so these measurements are made from electron microscopy. So you can see here EM images. This is a 2020 work, but the earliest connectomics work dated all the way back to the early 80s. And it allows you to map out every chemical synapse and every gap junction in the brain. So anytime two neurons have a connection, you can map that out, and this is what the map looks like. And so you might ask, well, we have this connectome. I mean, aren't we done? This is the wiring diagram. One of the things I want to stress is that even though this is the most comprehensive connectome of any organism, there are some things some information you can't get from the connectome. So the connectome is a static picture. This one's from one worm. We now have 4 worms worth of connectomes, or maybe a couple more now. So it doesn't have anything about plasticity. It doesn't tell you any temporal information. And even beyond that, it doesn't give you information about necessarily information about the signs of these connections. So in general, we don't always know whether a specific connection here is excitatory or inhibitory. And we have heuristics for estimating the strength, but they haven't been systematically validated on a large scale yet. Okay, so yes. That's a great question. Thank you. So for those on Zoom, the question was, what's the consistency of the connections across those 4 worms? So there isn't really an agreed upon metric. It's like one word metric that I can tell you. But the Meijen's group in this Whitflit paper were the first to image multiple connectomes. And the rule of thumb is something like this, that if you have 2 neurons that have many synaptic contacts between them, like 5, 10, something like that, you will consistently see many synaptic contacts between those 2 neurons across animals. But if you have very few synaptic contacts, then they may only exist in one of the 4 worms, for example. Okay, so that's the best I can convey. Did that answer your question? Yeah. And that'll come up, for example, because when we're doing some of our comparisons, we will look at that and say, look, this neuron, it has a connection, but only in one of the 4 worms, this probably isn't connected most of the time. And here, I want to evoke Eve Marder and Corey Bargman, who talk a lot about thinking about the connectome as kind of the menu. They think about this as the menu of latent circuits available to the nervous system, and that as you change state or as you add neuromodulators, you can turn on or off different ones, and so that these connections can change. Okay, but in C. elegans, we have decades of experience looking at this for our favorite circuits, like the touch circuit, and trying to, in a kind of retrospective way, write down these wiring diagrams and say, okay, this is really how the touch circuit works. But in our experience, you can do this if you've been studying a circuit as a community for maybe 30 years, but if you want to kind of look in a new place where you don't have decades of data to inform your intuition, it can be very challenging to trace out which paths matter. And so that was one of the motivations to do these measurements. Okay, and then I want to specifically point out some concrete cases where there are things going on that you don't see in the wiring diagram. So for example, there are known cases where you have a postsynaptic neuron that expresses receptors to molecules released by the presynaptic neuron, but they'll express both a excitatory or inhibitory receptor. And so this neuron is sending molecules that both excite and inhibit the other neuron. And so you're not going to be able to, even with all the genetic information, know what's excitatory or inhibitory. And then we also have known for a long time that neurons can communicate not along the wire. So they can dump synaptic, they can dump, yeah, go ahead, Bill.
So
Yes.
is this observation an exception to the usual? I don't know whether it's rule of thumb or anyhow, we were taught, right, that this doesn't happen.
Yeah, well, that's not,
Evidently,
that's false.
okay.
You're asking a more subtle question, which is how often is that false?
I mean, if you're not convinced that it's true for some reason, you stop.
Right. So,
Right, so this is 2011, right? So it's been 10 years.
but this was not the first.
So it's more than 10 years.
Yeah. So, okay. So I think so I don't have the answer off the top of my head, but we have, and I will show you, like Francesco's been building some neuroinformatics packages that allow us to scrape all of the amazing gene expression profile data to ask in principle this question, right? Like how often do you have, in principle, how often do you have at least gene expression of receptors for molecules with both polarities? Okay, so you might be surprised. It depends on what you're surprised about. Are you surprised that the presynaptic neuron is releasing two kinds of transmitters, or you were surprised that the?
That's right.
Well, but in C. elegans, one molecule can be, so for example, you can have a receptor can have both a receptor can have both inhibitory glutamate receptors and excitatory glutamate receptors. So there's not a one-to-one mapping from molecule to neuron response to begin with. But then it's also not true that one neuron can only release one kind of transmitter. And then it's also not true that one neuron has to have only one kind, either a excitatory or inhibitory receptor to the molecules from the presynaptic neuron. We now see evidence. Just, you don't have to take our word for it, like, very clear gene expression evidence showing all of that. You don't even need functional measurements, but you do need functional measurements at the end of the day if you want to know what is the net signal that the presynaptic neuron is giving to the postsynaptic neuron. And this is one of the rare cases where there was an electrophysiology recording. So you actually knew, and I think I took out the slide, but we recapitulate this using our all optical methods. And that's just for the stuff that runs on the wires, but neurons are also communicating by releasing molecules into the extracellular milieu. This often happens through dense core vesicles. It's most commonly associated with neuropeptides and monoamines, and that means that a neuron here can send molecules that diffuse over to another neuron, even though there's no synapse, chemical synapse or gap junction between them. We're often used to thinking about these as neuromodulatory, so acting on long time scales. Although if you talk to folks like Eve Martyr, they'll tell you, oh, well, actually, no, we think that back in the old days, we knew that these could be fast too. Okay, so our goal here is to measure responses to neural activation. We're calling this a signal propagation atlas. And so the name of the game here is we're going to stimulate neuron A, and we're going to measure the response in all the neurons, including neuron B. And in doing so, we're going to get empirically the strength of the connection, the sign, whether it's net excitatory or inhibitory, the causal arrow, temporal properties, and we're going to try to get something about the variability, and we're going to do this even for those connections that aren't wired. And there's been actually a lot of work over the past, almost 15 years now, developing methods for optical interrogations for measuring the signal propagation, including even early on in the worm, which I'm quite proud of. What's new here is that we're going to do this at brain scale and cellular resolution. And so there's a number of technical challenges that had to be overcome. So I'm just going to highlight 3 of them to give you a brief flavor. So one of the goals here is you have to target each neuron independently. We want random access to stimulate individual neurons in the brain. And we have to do this while we're imaging the whole population. And we need high throughput, so we have to do this automatically in closed loop. And so our team over there put together an instrument that does 2 photon stimulation. You need 2 photons so you can activate 1 neuron, even though all the neurons above and below and around express your actuator. And then developed real-time closed loop computer vision software to identify these neurons and automatically do these experiments. Okay, the next big challenge in this area is that it's very hard to find a pair of optical actuators and reporters that you can address independently. And so the team spent a couple of years going through different options and indicator pairs until we stumbled upon this one, this GER3 PRDX2 system discovered in the Horvitz lab, which is kind of a non-conventional actuator. But it's really nice because you can image your calcium indicator GCamp at 505 nanometers without activating any of your neurons. And you can activate this further in the purple or using 2 photons. And then to do the breadth here, we want to express our reporter and our actuator in every single neuron. We want a lot. And that makes for a very unhappy, unhealthy worm. And so there's another dial we have to play with, which is we want to get as much of these molecules into the worm with them being at least nominally healthy. And to do that, the team came up with this great system to skip over a lot of expression when we don't need it earlier in development and only turn on the expression of these molecules that we need to access the brain closer to when we do our experiments later in development. And so for that, we're using a drug-inducible system. Okay. Finally, there's another challenge that we didn't have to solve ourselves, thanks to our colleagues over at Columbia in the Hobart lab. We needed a way to identify the neurons we're recording from and map them onto the known connectome. And so this NeuroPal system converts the gene expression profile of the worm into a color code, a color barcode that you can read out, and that allows you to uniquely identify each neuron by its name. And then you can map that onto the connectome. Okay. So this is what the instrument that the team built looks like. The worm goes here. I added a little word that says worm, so it goes there. And this is what a typical experiment looks like. So you stimulate a single neuron here while imaging the rest. I've sped this up. You see that neuron respond in others, and then 30 seconds later, you stimulate another neuron, you see that neuron respond in some others, and you keep doing this one at a time for about 40 minutes. Okay. Yeah, Brad, what's up? The right is a Z projection. So it's showing you a different, it's showing you, yeah, exactly. It's showing you a different cut through the 3D space. Yeah, good question. And up on top, Brad, is, which I should have mentioned, is our reference. We report both the calcium activity, and we also have a calcium insensitive fluorescent reporter. So we know where all the neurons are, so our computer vision software can go find them and stimulate them. Yeah. We can, but we do that afterwards. So, or actually we do that before. We've done it both. Do we do it before now? We do it before. We do it before and after. Thank you, Sophie. So not while we're, that's what I should say, not while we're going and stimulating, but before and or after, we use separate optical instruments to measure the colors that are expressed. And that allows us to identify all the neurons. And then here, we just keep track through time of which neuron is which, so that then when we see that this neuron lit up, we carry that information through time, we keep track of that neuron and then look at its color and then relate it to the known identity. Yeah. In this case, we're stimulating for about 100 milliseconds. So these are brief and stimulations that are designed to be hopefully not so strong.
Is 30 seconds enough for the system to kind of reach a steady state of that?
That's a great question. This is something we're interested in and haven't measured super systematically, but by eye on this slide, you can get some sort of sense of how it decays back. And so 30 seconds was kind of our compromise between trying to increase the throughput by and giving this system some time to relax. I think there's a whole bunch to be done on different timescales, and I'm excited to think about that. Yeah. Yeah.
I'm trying to get a picture for the identification of the neurons.
Yeah.
I put different fluorescent proteins under the control of different motors, and so when the cell differentiating different on this combination of colors, if I have this common color.
That's right.
These colors overlap with, in order to get that one, they probably overlap with the range of which your dynamic measurements, right?
Well,
Maybe I'm missing something, but I don't.
so
If they're there, I would imagine they'd get away with it.
yeah, so so that's a great concern, and that was part of the design spec, basically. So the Hobart lab and our initial efforts and others who've tried to work on this, we've all tried to leave open the GCamp channel so that you have a clear channel to measure in the green, whether it's GCamp or GFP, because of course the power of these systems are to basically register or join data sets, right? So you want to be able to take your GFP line, you see GFP somewhere and you want to know where it is. So you want to be able to genetically cross it with the multicolor system. So you need to be able to see the green independently of all the other colors. And so there are 4, I believe, okay, my team can correct me on this. It's, I can rattle off, I think it's M orange, cerulean, BFP, and Neptune. CIOP and RP, yeah. So we've left the green open. Well, we haven't, the Hobart lab has. So these certainly overlap with our red, but then we don't care because it's just a static. We're not measuring any dynamics, and we just need something bright to see. From an instrumentation perspective, we are, when we're doing these kinds of experiments, we're really pushing the limits of what you can measure. We're imaging at 200 frames per second, 6 volumes per second. We have very brief exposures, so we're not splitting up our photons into color bins to try to measure all those faint colors. We do that offline. Yeah. So we do that the same way we do it here, actually. So we have a freely moving worm, and then we stop the worm from moving to do neuropal. So neuropal, more so than other techniques, requires either synchronous measurements of all the colors, right? So you can get the precise ratio, or in a mobilized worm, so you can take your time to image one color and then the other in time. So it would be more challenging to try to get all that color information in a moving animal where you have all these other constraints. Yeah, does that answer your question? But because we have developed very good tracking algorithms, we can do this in a moving worm, right? You track the neurons through time until the worm stops moving, and then you image all the colors. It's a great question. Thank you. Sorry. Okay, I'll remember to ask to repeat the questions for Zoom. Okay, this is what a single trial recording looks like. So this was recorded simultaneously. Here are all the neurons with their neuropal labels. These are the ones that we labeled or that we identified. And then on the top here are neurons that were stimulated at the time of their stimulation. And you can see we imaged for about let's see if I can make this move out of the way. We imaged for about 40 minutes. We imaged many neurons and we performed we typically perform order 80 stimulation events. And so let me just walk you through this. Here we stimulated neuron I3. You can see M3. You can see many neurons respond, including neuron M3L. And so if you look in the connectome, you would see that actually there are both direct and indirect connections between neuron I3 and M3. So you might expect M3 to respond. And every time we stimulate I3 and see M3's response, we can look both at I3's activity and M3's activity. And we do this across animals and sometimes multiple times within an animal. And so we can get information. So we get information across the population about how M3 responds to I3. And so here we've sorted these responses based on their amplitude, and you can see average traces here. So this is how signals propagate from I3 to M3, at least one metric. And one way we can represent that information is as a single pixel here here I'm showing the average amplitude of the calcium response. And the way you read this is down there, that's neuron I3 that we stimulated, and this is M3's response. And it's orange because the change in fluorescence was positive. And it's one pixel of many because we've done this for many neurons. So for all of these neurons we've stimulated and we've measured the network's response. So far, we're up to 43 animals, and we're about to image a whole bunch more. So this is one way of visualizing, but obviously we get more information than just the amplitude. Yeah, Brad, please. That's correct. That's a great question. So Brad points out that, we're in a particular state here. We have an immobilized animal that's not doing much. Can we look in different states to see how these change? And that is exactly one of the big motivations we went, for building this kind of system, was we wanted to catch the brain in different states and see how the functional connections change. I will not be showing you results of that yet because the first thing we have to do is do it in one state. But yeah. And I would love to, we have like this long list and it's almost overwhelming of where we go next. So I'd love to hear your thoughts, but we want to, yeah, exactly.
Distribution, mostly.
That's a great question. I have that on a slide, but I can tell you it's roughly 80% excitatory and 20% inhibitory. That's complicated though, because inhibition with GCAMP is a little complicated to think about. One of our interpretations here is we have slowly varying graded potentials. And so probably the inhibition we're seeing is the result of, slight tonic activation that then is inhibited. But it's interesting that people have done studies based on gene expression to try to guesstimate what fraction you might expect. And they also come up with 20%. So, that's always nice. Yeah.
I'm trying to think about the absolute scales of these things. So presumably when you flash the light, you know how bright it's spotlight.
Yep.
So that produces some amount of absorption by your actuator, which produces some amount of current.
Yeah.
So
No, go ahead.
Wait.
Go ahead.
Well, I don't know where in that signal which produces some voltage response in the neuron that you're actually recording from.
Right.
Presumably would drive the other. And so I don't know where.
That's right. So where do you?
So if I stimulate multiple times, each time I get a different response, this
Right.
where does that variability go potentially? If I want to add a small.
Thank you, Bill. So Bill asks, where do you, how do you think about variability? What is the right thing to measure presynaptically and postsynaptically? Because you have a laser intensity, but you also have the presynaptic neurons activity and you have the postsynaptic neurons activity. And so we've also been thinking exactly along those lines, thinking about what is the appropriate thing to measure. I think we we decided that one thing that we're really interested in is something about the transformation from the upstream neurons activity to the downstream neurons activity. And so one way we can try to capture that is to look at the upstream neurons activity and the downstream neurons activity for each instance and define a kernel, a linear kernel, such that when you convolve it with the upstream neurons activity, you get the downstream neurons activity. And so for every stimulus response event, now we have these different kernels, and we've put some constraints on how to construct these kernels so that they're, reasonably well behaved. And then we can ask questions about those kernels, how much they vary, what their timescales are separately. So previously, that wasn't what I was showing you. I was showing you just the average of the magnitude of the downstream response, which didn't have that information, right? Some of this could be variability from how much you activate the upstream neuron, but these kernels at least take the upstream neurons activity into account. Did that interest what you were asking, Bill? That's a great.
I guess if you do it trial by trial, the different kernel in your trial, you think about the average of those. That might be.
That's right. That's right. And in fact, Sophie has done some analysis, both to look at the variability of those kernels and to try to think about the average. The funny thing is, how do you average a kernel? I mean, you might have some better ideas than we did. I think, Sophie, one of the approaches you took that I thought was very elegant was you would basically in silico propagate activity through the different kernels and look at the downstream responses and average those. So you could see the average downstream response among all the different kernels. Yeah.
Much of this complicated by the existing spontaneous activity. Sometimes you get it on this already activity.
Yes. Great question. So how do you deal, how complicated is this? How do you deal with instances where you stimulate a neuron that was already active? Okay, so we have a couple things we're trying to do to think about that. One is we do try to keep track. So we have a layer of analysis so that if the neuron was, this neuron that's slowly decaying at this time point, we're not going to say it's inhibitory because we can write a simple classifier that says, look, this thing was decaying before. So we do some kind of data handling there. The other way we try to avoid having that be systematic in our data is randomizing the order in which we stimulate. But those were the two approaches. The right way the other thing you can do is space out the interstimulus interval really large. And we can do that, when we can do that on a case-by-case basis too. But there's a bigger question, which is you expect things going on in the nervous system on multiple time scales. So you expect that at some point you hit a neuron that changes the internal state of the animal in a big way, and then the rest of your recording should be different. And that is really interesting. And so for that, I think that goes back to Brad's question. We want to, in a more controllable way, pull levers to put the animal into these different internal states and measure the changes in the connections. Okay, these are great questions, guys. Okay, so one of the things we wanted to do with these measurements is make these measurements of the functional properties of these connections that we're measuring available. And so we have two resources that were made thanks to our amazing Princeton research computing team and are built up, in this case, on top of some existing software, including from Meijen's lab, this Nemonode platform. And this allows you to browse the measurements, the amplitude of the functional measurements and compare them to the wiring diagram. So here we have our measured functional properties in purple, and the wiring diagram is in black. And then here, those kernels in a way give you some crude linear approximation of the dynamics of the brain. So you can go in and, in principle, inject any arbitrary waveform anywhere to any node in the network, and then just run those through those kernels to see what you would expect the network to respond. And so we've made that kind of widget available too. And this gives you those temporal responses that are missing in this kind of picture. Okay, I just want to give you a brief example. Not many here are wormy people, but there's just a wealth of information if you're interested in warm neural circuits too. So, for example, there's just all these neurons that we suddenly have information about that people haven't really studied for a long time or maybe ever. So, you can just choose a spot on the map. Like here, this is this bright red spot. It's like, okay, so that's AVJR to AVDR. Well, what's up with AVJ? Like, I don't hear much about AVJ. So Bill Schaefer had a paper from more than 20 years ago, which was kind of the last paper that I found that looked at AVJ that says, okay, this is probably egg laying and forward locomotion related. But if you look, you see these nice big responses in AVD. And one thing that the community is very confident in is AVD has to do with backward locomotion. And so it's like, new hypothesis, right? This is like an interesting new finding. And so this is just a wealth of information for those kinds of circuit level investigations. And in fact, you can see why Bill thought it might be related to forward locomotion, because you have in the connectome, you have this big black connectome synapse to AVB, but functionally you get these big recurrent connections with AVD. So, anyway, okay. That's that. All right, so that was goal one, which is provide this resource for cracking circuits and for constraining models. Then goal two was to investigate how the nervous system structure relates to function, because we have this privileged position where we have the connectome and we have these functional measurements. And so we started with asking some simple questions like the ones you asked. Says, does this make sense with anything we would expect from the connectome? And so we find a lot of things that do make sense. So we would expect that bilaterally symmetric neurons to be more connected. They often have gap junctions between them. And we see that. So bilaterally symmetric neurons are something like, I don't know, 4 or 5, fourfold more likely to be connected than two neurons plucked at random. And I have to be careful here. I'm using the term functionally connected to mean we see a response of some kind when we stimulated it. Okay. I'm here showing that for some reasonable threshold, about 20% of the connections seem to be inhibitory. And then if you believe that the connectome matters at all, you should certainly hope that as you get more hops through the wiring diagram away, you should see less functional connections. It's a little tricky because in the worm, every neuron is connected to every other neuron in no more than 4 hops through the wiring diagram. But we see that the likelihood of being functionally connected, according to our criteria, decreases as the minimum length through the anatomical network goes up. So that all agrees. Okay, so then we have this challenge, which is if you want to compare structure and function, how do you bridge these descriptions that actually are fairly fundamentally different? When we stimulate a neuron and measure the response of another neuron, what you're measuring is the sum of all the paths through the network. So you're measuring contributions both of this direct path, what neuroscientists call monosynaptic, and all the polysynaptic paths. And you're getting effects like including recurrence. If you look at the adjacency matrix or the W or something, you're getting just the information about the weights of the anatomical connection between A and B. There's different ways we've thought about how you relate these. One of the ways that we thought was most principled would be to repeat our functional networks in silico in a biophysical model that uses the connectome as its constraints. And so that's what we've done. So in silico, we stimulate neuron A, we take an existing model that's fairly well accepted by the community. We take weights from here, and we take other information from gene expression. The weights come from the connectome, and we play this forward, and then we compare. And I would, if you're saying this is somewhat dissatisfying, we get all kinds of reactions at this stage from saying like, well, how could you possibly expect the connectome to have this information? In which I'd say like, oh, well, we want to check. So we do see some agreement, right? So if you look at, you repeat our experiment in silico and you ask what you would expect the change in voltage to be based on anatomy, and you break up our you break it up into neurons you expect to have, some kind of response and not. We see that, we see some statistically significant difference between the fluorescence observed in these two categories. But if you dig deeper, it's somewhat dissatisfying. And I just want to briefly talk about sample size and effect size. So, we're measured for each of these neural pairs, we have different number of measurements and we have different effect size. So we have differences in sample size and effect size. So here, this is bigger effects and here has, more sampling. And so we had to come up with some kind of statistical framework to talk intelligently about this. So we have kind of two bins that we run statistical analyses are. These things we have high confidence that they're functional connections because either we have a few examples, but the effect is huge, or we have many examples, even if the effect is fairly small. And similarly, we want to test for cases where we're pretty confident there isn't any functional connection because we have many examples of almost no response. Okay, in between, there's probably interesting real stuff too. We definitely see instances that look very variable or bimodal too. But here we're going to have to ignore that for the moment.
Sorry.
Yeah.
Because I'm not exactly sure this is separate from the model, right?
This is separate from the model.
But if I imagine that what an anatomical error is at least for these small amplitude simulations, it's linear current, but it lives on the connection. Right? Then you also measure linear currents between all pairs that are in the limit, you get all the pairs. So is it the case that there are enough actual connections that by putting a different kernel on every arrow, I can reproduce any combination of functional connectivity? Or is it the case that you actually try to do that and you see something, you'd be attaching as a monosynaptic response something totally crazy, so you know that that's not the case?
Okay, I don't actually understand your question, Bill. I'm sorry.
As you point out, you measure you stimulate neuron A and B, you also see the indirect path.
Yes.
Presumably the direct paths are described by some relatively simple impulse response.
Yes.
Right.
Sure.
The question is, by combining all of the direct, by attaching reasonable impulse responses to all of the direct paths, can you reproduce any combination of functional connectivity?
Are you asking, can you do something intelligent, like look at all the information you have to reconstruct the paths that the signal actually took?
Yeah, I mean, in some way, it seems like what you've done is a forward to say.
Yes. How do you solve the inverse problem?
People think they know the connections. Somebody built a model of what lives on every arrow.
I mean, Francesco, you should speak up. Francesco wrote a nice paper in PRL that talked about how you would explicitly infer the inverse problem.
Yeah, I mean, the problem that we have here is that the activities that we use are relatively small. And so if you to run the kernels like a diet, get the kernels in the diet paths of the diet paths, you need better temporal resolution that we have to get the fine difference in time scales. So we think that in our case we're a little underconstrained to do that fully, especially because if you really want to do it, then a little bit, and we're missing some areas, some neurons,
But it's some I thought, Francesco, it's some combination of, even if we had all of those areas, to solve this inverse problem, to start here and infer these properties from here, you need very high signal to noise, a combined very high signal to noise and temporal information. So you can look for shared time scales and the paths to kind of disentangle, right? Like the indirect paths you're convolving multiple kernels, and you should be able to see that as a slight change in that kernel. And I think it's that signal to noise, the combined signal to noise and temporal information that we're lacking. Is that right?
Yeah, that our response C. elegans activity is, and part of it is the genetic figure that we use. They're very long, and so you need very good signals. Basically, you need to have signal to noise the
And for the folks on Zoom, the question that Bill's asking is, why can't you go from here to here? How do you infer that if you actually have all of your measurements? Is that fair?
Yeah.
Yeah.
I mean, I didn't mean it with any reproach.
No. I mean, I think you showed it's quite possible in principle.
I mean, well, if the signal the network is there and you have information, yeah, it's possible. If you start assuming you have at least
Well,
some.
for example, it could turn out that maybe, so there's one matrix, which is direct connections, no matrix, which are your functional connections. And I could get from one to the other, but I might discover that when I do that, the direct connections look really weird. For example, there could be direct connections in places where there are actually no direct physical connections, or the impulse responses could be intrinsically, could be way too long, or I don't know.
I think in part of what we saw at the sparsity system, it's probably not putting constraints, but I don't know.
I mean, I think this would be a ripe area for thinking more about. Yes.
So we calculate the different parts, so you have two choices, right? You have the chemical signals and the elegans, the reductions.
Yes.
What is your view? So what are you actually testing here? Which powers and how we actually get out? Say you take neurons and solution that you know that there is a gap happening, then you take another ones, you know that there is only chemical signals. So how the kernels
In the simulations. No, in experiments. Yeah, that's a great. So the question is, empirically, if I understand correctly, how do the kernels you measure or the functional connections you measure depend on the modality of whether it's a chemical synapse or gap junction? So I'll show you how we've tried to address that question for extrasynaptic signaling, and which is we have these powerful mutants, so we can turn off all most of extrasynaptic signaling in the worm and then repeat our measurements. And so on our to-do list is to actually turn off systematically gap junctions, for example, and see how the connections change. That's one empirical way of getting at that question. Another would be to do a more data-driven approach, and I'll show you one of Francesco's data packages that allows us to try to do. But I guess the short answer is we don't have an answer yet. Yeah.
This I don't mind help you cite the directionality that functions, because we know there's systems of quite a lot of functions.
Yeah, absolutely. I mean, Francesco, you just ran some of that data, and so Bassett, a talented undergrad, has also been looking at that. I don't know that we have anything we can say at this point. Is that fair?
Yeah. For now, I don't have a good answer.
But high on the list. Good idea.
There's a lot of things. To be sure you're talking about that, connection, then it shouldn't be have to so that is
So for Zoom, the question was, do you see evidence of rectified gap junctions in your data set? And the short answer is, we're really excited about that question and looking at it. Thanks. Okay. All right. Good thing I went with the short tack. Okay. So, so we did do this play it forward biophysical model that allows us to at least compare our measured responses on the y-axis to what you would predict on the in voltage from the biophysical model. And what we can say is, these things don't agree very well. And there's probably a bunch of reasons for that, some more interesting than others, many of them probably on the modeling side, which is that, making the assumptions that go into these biophysical models may not be correct, right? You're taking conductances and parameters that were measured for one neuron and applying them to all the neurons. But one of the things we can say that was pretty interesting was, if you get rid of extrasynaptic signaling, you disagree a little less. And so this is a mutant that's defective that has defects in dense core vesicle release in the docking and priming of dense core vesicles. And so that should disrupt a lot, most extrasynaptic signaling. And so for this last little part of the talk, I was going to talk about one of our findings, which is that extrasynaptic signaling also seems to drive brain dynamics. And I'll show you some evidence of that. So we started with this one neuron RID, which is kind of a famous neuron in the literature because it's supposed to signal extrasynaptically. There's no direct paths from RID to these other neurons here. And we know the right peptides are expressed, and it had been well characterized, but nobody had measured it functionally. And so what you can see is here I'm just showing you this neuron's response to RID stimulation. You can see all these neurons respond to RID stimulation in wild type, but in the UNC31, the response is much lower or sometimes goes away. But it's not like this mutant breaks all responses. Here's our mutant that's defective for extrasynaptic signaling. If you look places that should have gap junctions between them, for example, you still see responses even in the mutant. So we think we've identified for that neuron pair a dense core vesicle mediated response that's likely peptidergic. And so we went ahead and did a kind of screen to look for instances of purely extrasynaptic signaling, or I should say like purely dense core vesicle dependent signaling. So we looked for hits that have responses in wild type, but not in UNC31. And so I have a cheeky graph here showing the top 75. We show that on average, our biophysical model suggests that there should be much less responses for this population, right? So these things over here are like reasonable responses, and there's much less of them. You can look at the cumulative distribution function in our hits. So anatomy would suggest these things shouldn't be connected, but then when you go look, you see these kind of really cool connections. So AVJ, activating AVJ gives responses in ASG, but that goes away in the mutant. And when you go look, there's been some really exciting work in the field just in the past few months to look systematically at the expression of these of neuropeptides and to try to identify which GPCRs respond to which neuropeptides. And when you put that data together, you find that this downstream neuron expresses a bunch of GPCRs, a bunch of receptors that respond to a bunch of peptides produced by this neuron. I mean, so this makes sense and is consistent. And we even see things, this is kind of more for the wormy crowd, that I found very surprising, which is here we see bilaterally symmetric neurons communicating with one another via peptides. So people have known that there can be extrasynaptic peptidergic autocrine signaling where a neuron class releases peptides that activates itself. Here, AVDR and AVDL, even though they're bilaterally symmetric, and so in the worm field, they're often thought of as one thing. If you actually look, they don't have, only one of the four connectomes has a gap junction between it. So often it has no gap junction between it. And in the mutant signaling, you can stimulate AVDR and there's no response in AVDL, only in wild type. And so this is an example of extrasynaptic peptidergic signaling, even between bilaterally symmetric neurons. And then the last data slide I have for you is that the signaling can also be fast, at least fast by my naive prior. So, we're somewhat limited in our temporal resolution, but this bar here is half a second. And if you compare the neurons that seem to be signaling extrasynaptically, and I think this gets at your question, which is, here we've identified signaling happening of a specific modality, right? This is not gap junctions or chemical synapses. This is extrasynaptic signaling, and yet it can happen on timescales, at least with our resolution, down to half a second. Yeah.
That's where you're not.
So this is a very good question. So in the C. elegans connectome, near and far is a little subtle because you don't, if it's extrasynaptic, you don't know where it's actually releasing, right? And the nerve, you have these neuron bundles basically, and so you can find neurons that have cell somas that are very far away, but very often their neurites might be coming within very close to one another. We did do the back of the envelope calculation, which is, if I release I have a peptide of a given size, what's the diffusion coefficient, what's the Reynolds, what's the viscosity, how long would it take? And you basically can get a molecule the size of a small peptide from anywhere in the brain to anywhere in the head to anywhere else in the head in less than a second, because it's small, right? It's only like 30 microns in this dimension and maybe up to 70 in the other dimension. That brings us to another point, though, that I'm going to hit in 2 slides, which is the worm just special because it's so small? I guess, okay, sorry, I lied. There's one more data slide here, which is we also wanted to reach out to spontaneous activity, right? So far, I've been comparing our functional measurements to the connectome, but you could say, well, how well do our functional measurements recapitulate the kind of spontaneous activity you might see? And so we measured a correlation matrix of spontaneous activity from an immobilized worm not doing anything. And we've done some kind of analyses here to show that under certain assumptions, we agree much better at with the correlation matrix from a spontaneous activity than if you just use the connectome itself. And I can talk about that. Okay, so I presented a functional atlas of the brain measured by neural activation at single cell resolution. Functional connectivity differs from anatomy, at least our simple models used in anatomy, in part because it emits extrasynaptic signaling in the brain, and that we're pretty confident on because you can't see that from anatomy, and we see that in functionally. And we see that functional connectivity better, okay, I showed you that functional connectivity better predicts spontaneous activity than anatomy. And one of the things that I found fairly striking is that neuropeptides seem to be another option for the brain for signaling on these less than half-second timescales, which means they could directly contribute to dynamics, whereas I often had this prior that neuromodulators are going to adjust the weights of the brain over very long timescales, like 30 seconds, minutes, that kind of thing. And so, this raises all these questions. We want to look at how functional connectivity changes with learning, with neuromodulation, and with all these internal states, as I think one of you asked. And it prompted 2 other questions I'll just mention, which is, given that I think for the foreseeable future, experimentally, we're going to have connectomes before we have comprehensive functional measurements. How can we do a better job inferring these functional properties from the connectome and from other measurements that we might get sooner, right? Because what we did in the worm is still many, many years away from being done in higher order organisms, at least on the same scale. And so one idea is that we need better data sets to constrain our models, and we ought to be able to do that by pooling different types of information. And so Francesco made this Python script, this Python package, the Worm Neuro Atlas, that combines a bunch of different data sets, like from the single cell gene expression profile from SenGen, the connectome, the genome, our signal propagation atlas, and these de-orphanization studies into a useful package so you can do neuroinformatics questions like looking for those rectified gap junctions, for example. Here's one, Bill Schaefer had this amazing paper that's really impressive showing how trying to predict these wireless connections using the gene expression profile and the deorphanization data. And now you can do this in a couple lines of code in Francesco's package. So it makes combining these data sets really straightforward. So, as I think about the future of modeling, it will be to leverage, to constrain not just with anatomy, but with these kinds of additional data sets from gene expression and deorphanization studies. Okay, and then last question. So is the worm unique, right? Well, we know it's kind of unique. It's pretty small. You have only 302 neurons and it's, order 50 microns length scale. As you move into other organisms, you're spanning many orders of magnitude. And so one of the ways we are going to try to answer that experimentally that we're just talking to is we're starting to talk to our colleagues down the hall to ask, hey, look, you guys just measured a brand new connectome. Mala and Sebastian have this very comprehensive connectome from the Flywire project that has over 120,000 neurons fully proofread. We're now looking in there to see, well, how often do we see dense core vesicles in the connectome that might indicate extrasynaptic signaling? And we can't obviously do this on, we can't obviously measure systematically 120,000 neurons anytime soon, but we could at least choose some subcircuits and use optogenetics and whole brain imaging in those subcircuits to try to see what the functional connections look like and whether extrasynaptic signaling is present. Okay, so with that, I want to thank my lab, many of whom are here today or on Zoom, and want to thank my collaborators and the funders. Yeah, there's the lab. Thank you. That's great. We got lots of great questions during the chat, during the talk. Let me just see if there's any on Zoom. And if not, we can all do coffee. Gautam. How good are we at doing whole brain imaging and freely moving worms? Yeah, well, I like to think we're pretty good at it. Our group was one of the first to do that. And most of our measurements in the lab are in freely moving animals. We had to compromise for this kind of data set and do it in the mobile animals. I don't think I heard the second part of your question. The first part for Zoom was, how good are you at measuring in freely moving animals? What was the second part? Yeah, absolutely. Absolutely. And in fact, so Gautam asks, could you integrate the calcium activity you see in your freely moving animals with the functional measurements here? And that's exactly something that I think Francesco, you and Tori and others have been thinking about, which is, could you explain the dynamics you see in a freely moving worm with these kernels that we measure? But I don't have results for that today.
To what extent have you thought about, or do you try to look at nonlinear interactions of neurons with this type of experiment and maybe specifically what kind of things the data detection?
Yeah, that's a great question. So the question is, to what extent can you measure nonlinear interactions? So Sophie, you're working on this right now. So we don't have any data to show you, but the first step is to look systematically in some small circuits where we activate 2 neurons at once and compare it to 1 neuron. Yeah. The problem, obviously the perturbation space explodes, right? You could look at all the pairwise, but now it goes as n squared. And then if you want to look at different intensities and nonlinearities, so, but on small circuits, I think we could do a kind of comprehensive investigation. Great. Thank you so much. Thanks, Bill.
For our last talk today, it's a pleasure to have Braden Brinkman here, all the way from Stony Brook. Braden's worked on change theoretical physics, worked on a variety of problems, particularly in dynamical problems in neuroscience. I think for those of you who follow the theoretical literature on models of neural networks, you will know that most of the neurons in those models don't spike, which is interesting, because most of the neurons in your brain spike. You should worry about that. And so it's great to be able to produce somebody who's going to talk about networks of spiking neurons. And I think that for a long time, many people in the community have shared the intuition that renormalization ideas should be useful in thinking about the nervous system, but actually delivering on that is hard. And so, Braden.
All right. Thank you for the introduction, Bill, and thank you everyone for sticking around all the way to a Friday afternoon, which is going to be capped off with something more abstract than the first two talks. So hopefully, I'm going to try to start somewhere near the beginning to try to get everybody on the same page, at least at a high level. We'll see if I was too ambitious in terms of how much I'm going to try to get through. I should remember to turn this on for the people in the audience on Zoom. So this is the More and More Neuron Symposium, and so we've looked at measuring from lots of neurons in a system and dealing with tens of thousands of neurons and these big data analysis problems. The way I come at this question as a theoretical physicist is from this idea that was put forward a long time ago by Phil Anderson, which is this idea that more is different. That when you have a lot of elementary things interacting together, even if they're quite simple, simpler than even single neurons are, you can get sorts of complex kinds of behavior that you might not guess would be possible by just looking at one of those single elements. So for example, if we want to talk about ice or the flow of water, that involves the interactions of many H2O molecules. You can't necessarily guess things about ice or liquid water or gas from just looking at the properties of a single H2O molecule. And similarly, we like to think that to understand the sorts of computations that go on in the brain and things like sensory processing and cognition, while single neurons may be doing many interesting things, we may need to understand what's sort of going on at the sort of collective level of coordinated interactions between neurons. And when we talk about that level, practically speaking, it's going to be very difficult to describe single neurons in detail. And so, in the tradition of physics, we tend to move towards these statistical descriptions of the dynamics of the system and trying to talk about collective behavior, and in particular, possibly transitions between different kinds of collective behaviors. And this is where these ideas from this thing called the renormalization group come into play, which was sort of put on a firm footing by Ken Wilson back in the 70s. And I'll just take a very brief second to distinguish between the renormalization group and this thing that you may have heard of in popular science called renormalization, which is usually talked about as some sketchy procedure for sweeping infinities under the rug. So they're related, but the renormalization group is the way that condensed matter physicists like to think about the interactions between degrees of freedom of a system at different length scales. And this renormalization group idea was profound, and the way we understood many different kinds of phase transitions and collective states of matter in physical systems. One that you're all assuredly familiar with is the transition between liquid water and its gaseous state. So these are two different phases of water, at least at a particular pressure or set of pressures. And there's as you all know, there's a boiling point. So when you heat the water to a particular temperature, there's a transition from liquid to gas. And it turned out, and one can show through this renormalization group idea, that this transition is actually intimately related to a transition that you would see in a magnet. So if you take a magnet, like a regular bar magnet, so you know that you can pick up paper clips with it, you may not know that if you heat that magnet up, eventually that magnetization is going to vanish. And that bar magnet would not have any sort of magnetic properties anymore. And through the renormalization group, one is actually able to show that these two transitions are similar in several very profound ways. So, for example, if you look at the liquid and you plot a particular difference in densities as a function in the temperature, you would get a plot that looks something like this, where it's non-zero, and at a particular critical temperature, that density difference vanishes. And if you plot something very similar, but for the magnetization of that bar magnet, you get a plot that looks very similar, where you have some non-zero magnetization, and then at a particular critical temperature, those things vanish. And if you look very close at this transition point in these two systems, what you find is that they scale like a certain power of the difference between the temperature and the critical temperature. And one of the big surprises of physics back in the early 1900s was, first, that this exponent turned out to not be something nice like a half. So a half is the value that you would predict by something called mean field theory, which I'll talk about a little bit shortly. So it turned out that this value was not a half, it's something closer to 0.32 in 3 dimensions. But the other surprise was that if you measure this transition in the liquids and you measure this transition in the magnets, these values turn out to be exactly the same. And so this established a notion of a universality class, that certain types of phase transitions are in the same universality class, and there are certain properties, like these exponents, that are shared by all systems in those universality classes, despite the fact that you might be talking about things with very different microscopic details, like liquids versus magnets. And the renormalization group was how we understood where A, these non-trivial values came from, and B, why seemingly very different systems fell into these same universality classes. And so what I want to talk about today is sort of an application of this renormalization group idea to spiking network models to try to get at understanding where some of this potential universality might come from in neural systems. And so I wanted to motivate with a particular, or what is believed to be an example of this in neural systems, which is something called neural avalanches. So the idea behind an avalanche is that, let's imagine for a moment that we're looking, say, at a slice of neural tissue that's not super spontaneously active, and every now and then, either the experimenter delivers an impulse, or maybe a neuron gets hit by a cosmic ray particle or something that causes it to fire a spike, and then it's going to send that pulse of electrical activity to its postsynaptic targets, and that's potentially will cause them to fire spikes as well. And so you get this cascade of activity that may eventually peter out. And if it peters out, we would call this a subcritical piece of tissue. So if you look at the distribution of, say, how many neurons fired or how long it took before this cascade of activity died out, what you would typically find is that this distribution on a log-log scale would sort of get cut off exponentially. So that's what this would look like on a log-log scale, and this could be the size distribution or the duration distribution. So this would be the frequency of sizes and sizes on the horizontal axis, for example. And this is what you might expect if you have too little excitation in the network. Now, if you looked at a slice of tissue, though, where those excitatory synaptic connections were extremely strong, what you might get is avalanches that do not peter out, but then they end up spanning the entire piece of tissue. And maybe this would be something like a something like a seizure where it spreads across the entire piece of tissue. And so if you look at the size distribution, you might get some small avalanches, but then you might also get this big cluster of avalanches sort of at the size of the circuit that you're looking at. And right in between these two cases is something that we would call the critical regime. So this is sort of the balance point where the strength of those synaptic connections is just strong enough that basically on average, each neuron is able to trigger one other neuron in the network. And what that leads to is, at least theoretically, is a power law distribution of event sizes, either the number of neurons recruited in an avalanche or even the duration of these avalanches. And so this is an example of a non-equilibrium phase transition. So different than the phase transition from liquid water into gas, which would be an equilibrium phase transition. This is an example of a transition where we are well out of the equilibrium regime. And it's been put forward something called the critical brain hypothesis, which posits that the brain regulates some of its neural circuitry to be poised at least close to one of these phase transitions, typically perhaps in the subcritical regime, but close to the critical regime. And today, I don't want to come down one way or another on the idea that the brain is actively regulated close to one of these critical points. But what I want to do is develop this renormalization group formalism to apply to networks of spiking models that are commonly used in neuroscience. So these ideas of the renormalization group, which I'll explain what that is soon, they have been applied in neuroscience before. Bill's applied them in many of his recent papers, but a lot of it's been either sort of at at the level of data analysis, or when it's been applied to theoretical models, they've often been models that have been borrowed from statistical physics and reinterpreted in a neuroscience context, but they typically don't involve spikes. They're typically, you could maybe think of them as involving coarse-grained states of neural activity, so active or inactive states. But to my knowledge, they've not yet been applied to things like leaky integrate and fire models, which we commonly use to try to model spiking activity in neural networks. And so that was one of the motivations was, can we develop an analysis that applies to spiking network models? Now, to give some specificity for the sorts of things that we might try to look for if these spiking networks are critical, is things like these universal power laws. So if we look again at the distribution of avalanche sizes or the distribution of avalanche durations, we might expect that if the network is at a critical point, they have some power law scaling. In any real tissue, you're going to run up to against finite size effects, but there would be some regime where you might see power law scaling, and we would expect that these exponents are going to be these so-called universal quantities, meaning that they're not going to be dependent on the microscopic details of the connections, but some sort of higher level properties. And I'll explain in a moment what we typically expect those higher level properties to be, at least in the physical systems that condensed matter physicists would study. And we can also look at more complicated things than just probability distributions. So for example, if you measure the average avalanche as a function of time conditioned on the duration of the avalanche, one can write down a scaling theory that predicts that the amplitude should basically scale with a particular power of the duration of the avalanche. And then you could do a kind of Whittem scaling collapse to collapse all of these curves onto a single curve. And so as I mentioned, things like these critical exponents and even the scaling function that's labeled F here, these are things which are predicted to be universal in the sense that we shouldn't need to get the details, incorporate all the details of things like ion channels and so on to be able to predict these numbers. And so that's that's why these are quantities that physicists would tend to like, because we can get away with writing down what otherwise seem like stupidly simple models, but we still have some hope of predicting what these values would be. And another point of interest is that the data that I'm showing here is from a 2012 paper analyzing neural avalanches. And what they found were values of these exponents, which, similar to the case of the liquid and the magnet that I talked about here, deviated from the predictions of mean field theory. And some of you may be aware that mean field theory is often described as basic what you would get from dimensional analysis, and that these deviations from mean field theory, sometimes it's described as a breakdown of dimensional analysis. Really what it means is that there is a dimension that you missed in your dimensional analysis, which allows you to get these very non-trivial values. And so we want to understand that if we're in a regime where we're getting non-mean field exponents, what properties of the network, say, are contributing to these non-mean field values. And so I want to try to provide some partial answers to some questions today. One is the example that I gave so far was what I would call maybe what happens in vitro, where you have very low spontaneous activity in the network. But if you look in cortex, as we saw in the previous talk, cortex can be very spontaneously active. And in that case, you may not be able to, it may be ill-posed to try to separate these different avalanche type events. And we might wonder, if there is a phase transition in very spontaneous cortical networks, might it be very different than the type of phase transition we would see in slices, which is, often the example is these neural avalanches. And then the second question I want to try to give an answer to today is, what is the appropriate dimension of a network? And by that I mean that, and the relevance of that is that these critical exponents do depend on the spatial dimension. So this is the value that you would get in a three-dimensional magnet or a three-dimensional liquid. If you looked at a two-dimensional film, these values would be 1/8. And so we know that dimension is one of the things that determines these critical exponents, but it's not clear what the appropriate dimension is for a neural circuit. Is it the spatial dimension in which it's embedded, or is it something like one of these fractal dimensions that people have defined for networks? And so that's one of the questions that I want to answer, at least for symmetric networks. And then the other question that I want to provide an answer for today is, there's a term that you will see sometimes in the physics literature, which is this long wavelength physics. And that's referring to the fact that to understand this sort of behavior, the scaling behavior, what we're looking at is sort of the behavior of a collective state of matter at large length scales in space and time. And it's not clear that that is the sort of appropriate coarse-grained degree of freedom for a neural network. And typically the reason that long wavelengths in terms of say momentum states is useful in condensed matter physics is that you often have things like crystals or translation invariant lattices, which makes that sort of description of the system useful. But that's not necessarily going to be the case when you have a random network. And so we want to know what is the appropriate basis for a more arbitrary network. So I will cover both lattices and more random networks in this talk. And then, as I mentioned, I may be too ambitious in what I want to get through today. So if you want all the gory details, there is a recent gigantic preprint. That is the identifier. And so all the technical details that I'm going to skip for the sake of time will be in that preprint. All right. So I want to start off, hopefully that gives everyone some idea what's gonna happen. But I want to start off by just telling you what the spiking model I'm going to use here is, so that we're all on the same page with that, and then also give a couple of examples of the types of phase transitions that we might expect in this model. So the spiking network model that I'm going to be working with today is something called the soft threshold leaky integrate and fire model, or the stochastic leaky integrate and fire model. So this is a model in which we have a bunch of neurons connected by some synaptic connections, and we're recording their membrane potentials and their spiking activity. And so what happens in this model is that if some neuron fires a spike, it sends that spike along its synaptic connection to its postsynaptic targets, and then that's going to cause some deflection in the membrane potential of the postsynaptic neuron. And here we're assuming this is an excitatory connection, so it increases the membrane potential. Now, in the more common leaky integrate and fire model that many of you may be familiar with, neurons spike in that model when their membrane potential hits a particular threshold, and that is a deterministic event. In this model that I'm talking about today, instead of making spiking completely deterministic, we're going to assume it's probabilistic, so that the probability that a neuron fires a spike is going to increase the more its membrane potential increases from the resting level. So there's going to be some firing rate nonlinearity that I'm going to call phi here, and the more that neuron's membrane potential increases, and I've put the nonlinearity on its side, so this is the vertical axis here is membrane potential, and the horizontal axis would be the firing probability. So the more input that neuron gets, or the more excitatory neuron that input gets, the higher the probability of firing in a short time window is going to be. And so if we write down the actual system of equations for this, the membrane potential dynamics are just leaky dynamics. So we have the membrane voltage in V, the rest potential, which is this curly E, a membrane time constant. And so basically what would happen from this part of the dynamics is just if the membrane potential is above its rest level, it's going to decay back towards the rest level. Similarly, if it's below the rest level. And then we get the synaptic input to the network. So this N dot here are the spike trains from the presynaptic neurons. So I here is labeling some neuron in the network. And so N dot J here are the spike trains from other neurons in the network. And they're providing synaptic input that gets weighted by these synaptic weights Jij. So every time a neuron fires a spike, it's good. Well, let's say some neuron 2 or neuron 1 in this example fires a spike. It's going to increase the membrane potential of neuron 2 by some value J21. And then, as I mentioned, the probability that the neuron fires a spike depends on the membrane potential and this firing rate nonlinearity. And in this model, the way we're going to quantify it is we're going to say that the total number of spikes n dot dt in a short time window dt is just distributed Poisson or Bernoulli here at this firing rate nonlinearity phi, which depends on the current value of the membrane potential. So the spiking is conditionally Poisson, but it's coupled, ultimately it's non-Poissonian due to the coupling with the history of the membrane potential and the spiking dynamics. So are there any questions about the model setup so far? Mostly for technical reasons. So the formalism, the mathematical formalism that I'm going to use would be extremely difficult to implement with a regular deterministic leaky integrate and fire model. It would be great if we could also do it for the pure deterministic version, but this is the one where we can actually do the calculation in the first place, and we'll view it sort of as perhaps a phenomenological model of spontaneous activity in cortex. You could imagine writing down the deterministic leaky integrate and fire model with just a like noisy current input, but that's much more difficult to work with in the path integral formalism that I'll touch on later.
I think about the usual integrated fire model, I have a reset.
Yes.
And so action potential is occur when voltage reaches threshold and then you reset it.
Yeah.
So here, they occur probabilistically, but you also don't have to.
So you can implement what I would call a soft reset by allowing JII, the connection of a neuron, onto itself to be a negative value. So in some of the results I show, I do implement the reset that way. In some of the random networks, for simplicity, I leave off that soft reset because it doesn't really change anything as long as that reset value is not too large.
If I let the non-linearity if you let the non-linearity become a step function, put JII at just the right value, is that the deterministic integration?
Almost. So the question for everyone on Zoom was, if I make this nonlinearity a step function, then is it more like the deterministic leaky integrate and fire model? And the answer is formally, but you would have to take the height of that step function to infinity. So in that case, basically, then yes, when the neuron gets over a certain threshold, when the membrane potential gets over a certain threshold, then you're guaranteed to fire a spike because that nonlinearity diverges. I've not tried to formally investigate that limit because it gets tricky, because then you have to worry about orders of limits and things. Any other questions?
Would this model be equivalent to a deterministic one in which the threshold is distributed in the same fashion as this one?
So the question was, would this model be equivalent to a deterministic model with a distribution of hard thresholds? I do not know. I could imagine that if you did, there may be an equivalence, like if you average over the distribution of thresholds, then maybe you would get something kind of like this, but I don't know. Anything else? So, I mean, part of the point of doing the calculation is trying to find out what details do actually matter for these so-called universal quantities. And I will try to point out one where, so for example, one of the details here is that notice that the membrane dynamics here themselves are linear. And so later I'm going to formally solve for the membrane potential. So in this model, the membrane potential is basically just a linear filtering of the spiking dynamics. And I'm going to consider that case because it gives us some theoretical guarantees for some of the calculations that I'm going to do. You could relax that, and it would change at least one of the critical exponents, which will have some additional effects. But part of the point of doing this is being able to identify, what details will actually change some of these critical properties. And so maybe the distinction between a hard threshold model with some heterogeneous inputs that makes it look noisy would ultimately not be very different from this model where we assume stochasticity from the outset. But we'll have to do that calculation someday. Yes. Yeah, yes, and so we'll come to that. All right. So just to give you a sense of how different types of phase transitions might occur in this model, the way one typically analyzes this is through something called mean field theory. And what I mean by mean field theory here is that we're going to neglect the stochastic fluctuations. And so what that means in terms of the dynamics that I just wrote down is that we can replace that spike train with its average firing rate. So replacing that n dot with phi here. And then because I just want to sort of give a sketch of the types of phases that we might get from this network model, I'm going to show the case where we have an all-to-all network where every Jij is equal to J over N, but I want to stress that this is just going to be for illustrative purposes in this introduction. Later, the Jij is going to be either from a lattice or a Gaussian random network, and in the preprint, I also showed some stuff for a random regular network. So in particular, I wanted to show what happens if we have a transiently active versus a spontaneously active network. So we might model a transiently active network by choosing a rectified nonlinearity, as shown on the screen here. So here we have the membrane potential nonlinearity, and you see that below a certain value of the membrane potential, the firing rate is 0, meaning that if that neuron's membrane potential is negative, it will not fire, guaranteed. You can also work out then that if all of the neurons have membrane potentials below 0, none of them are going to fire. And so in the absence of any external input, the network will just stay quiet. So that's why this is called transiently active, because even if we start the network off with some activity, we don't necessarily know if it's going to be strong enough to maintain that activity. And so we can actually work out what the steady state of this network might be. So that steady state would be where this time derivative is 0. And so those of you familiar with nonlinear dynamics will be used to looking for intersections between V and the firing rate nonlinearity. And so what I plotted here in black is V divided by the strength of that synaptic connection. And I've chosen the rest potential so that, that's another thing that you have to tune, but I've set it up so that there's one intersection between V over J and the firing rate nonlinearity. So that fixed point is that intersection point represents the steady state of this network. And in this case, there is only one intersection at V equals 0. And so what that would tell us is that even if we start this network off with some non-zero level of activity, that synaptic connection is not strong enough for the network to maintain that activity, and the activity would eventually die out. And we can work out how quickly it would die out, and it would basically decay exponentially with a time scale that's the inverse of 1 minus J times the gain of the nonlinearity, which is its slope at 0 input. Now, you might wonder, well, what happens if 1 is exactly equal to J times the gain? In which case the coefficient of the time would vanish, and well, you can work that out, and that's exactly when you're at the critical point, and it turns out that at that point you would get what's called an algebraic decay of the membrane potentials to this steady state value, meaning that they decay as a power law at long times. And if we assume that the next leading order nonlinearity of this firing rate is quadratic in the membrane potential, this decay would look like 1 over the time. And I'm going to generalize this power law decay, so I'm going to introduce an exponent beta because later we're going to try to calculate this exponent, and we'll see that it will differ from 1 when we incorporate the effects of stochastic fluctuations. And then lastly, if you increase the synaptic strength even further, so J is increased, now we get 2 intersections of V over J with the firing rate nonlinearity, and you can show that that intersection that's higher up is actually the stable one. And so what that represents is a steady state of the network where the network is able to self-sustain its own firing rate activity. And so you would expect the network to stay active when the synaptic connections are strong enough. And so this is maybe a model that could describe those avalanche dynamics, so that if if there's no input to the network and the synaptic connections are not too strong, you hit it with some input, it's going to cause a spread of activity, but that spread of activity would eventually die back out, at least as long as those synaptic strengths are not too large. Now I want to contrast this with, So, yes, so in the exponential case, this would really be this factor times T over tau. In the critical case, the tau falls out. So I've done the physicist's bad habit of at some point setting a constant equal to 1. So my time units are in units of tau. All right. So I want to contrast this with what might happen in a spontaneously active network. So the nonlinearity for a spontaneously active network, one would typically choose it to be something like the sigmoidal. And what I'm plotting here is the nonlinearity minus its baseline values. So phi of 0 would set the baseline firing rate of this network. And so here we would get stochastic behavior no matter what the neuron's membrane potential is, although if its membrane potential is very negative, that's going to suppress its firing rate. And in the particular model I'm showing, that would be the limit in which you would get a very low amount of activity if the membrane potentials are all very negative. And so we could do the same sort of analysis, and if the synaptic connections are not very strong, there's just a fixed point at v equals 0, and this would just basically represent the state where the neurons are firing at their baseline firing rate, so think of it as asynchronous activity. And you could work out what the decay, if you start the network with some neurons away from that steady state, they would decay again exponentially back to it. And then there's a critical value of the synaptic strength J, where they would decay very slowly back towards that critical point. And in this particular example, I'm showing a sigmoidal nonlinearity that's symmetric across the vertical axis. So the leading nonlinear behavior would go like v to the cubed. And so in that case, what we would expect is that the speed of that power law decay would actually be 1 over the square root of the time at large times. And I'm showing that I'm choosing that example in particular because, as was mentioned earlier, often we expect the symmetries to be the things that determine these critical exponents. But this is a case in which this symmetry that I'm showing with this nonlinearity is actually not protected against stochastic fluctuations. And so we're going to see that this mean field prediction for this particular model will be invalidated at that critical point. And then lastly, we can also look at the supercritical regime where the synaptic strength is now very large. And then we would get 2 fixed points that you can show are stable. And one would correspond to like a high firing rate state. The other one would correspond to a low firing rate state. And so this looks a little bit like, maybe something like the Ising model, which some of you may be familiar with, that we have these 2 metastable states. And so you might think that this is in the Ising universality class. And I'm going to hopefully get to later that the picture is more complicated than that due to the effects of these stochastic fluctuations coming from the spiking activity. So that was the mean field picture. So that was what we would expect on the basis of neglecting these stochastic fluctuations. We can show for this model that if you wanted to look at the dynamics of the actual expected values of the membrane potential. So if imagine we're doing a bunch of trials and you're averaging over those trials to estimate what the average membrane potential would be, it turns out that the dynamics of the expected values of the means would look similar to the mean field model, except that you have some effective nonlinearity that I've highlighted in red and labeled capital phi here. So in the physics lingo, we would say that firing rate nonlinearity has been renormalized by these stochastic fluctuations. And one of the goals of the formalism that I'm going to talk about today is to try to estimate what that nonlinearity would be, how it would change from the bare nonlinearity, and what implications that might have for the collective behavior and possible phase transitions in this network. And so first goal, the goal of the first part will be to calculate that effective nonlinearity. And then the goal of the second part will be to get back to these so-called universal properties and trying to estimate those and say something about universality classes in the network. All right, so to get to that first goal, now I'm going to give sort of the high level idea of what this renormalization group procedure is and how we can use that to calculate that effective firing rate nonlinearity. So the formalism here, the setup, involves something called statistical field theory. So what that means is that imagine we have a probability for all the possible membrane potential traces that you could measure and all the possible spike trains that you could measure. It turns out we can write that formally as an integration over some auxiliary variables that I'm going to call response fields times a factor that looks like e to the minus s, where s is something conventionally called the action, and it encodes the dynamics and the fluctuations of the model. So you could think of it basically the action loosely like the log probability, but in an expanded space where in addition to the membrane potentials and the spike trains, we have to introduce these two extra fields that I've labeled with tildes, which are conventionally called response fields because they're related to how the network is going to respond to perturbations. So we have these response fields, and then what I'm showing in orange here is just the membrane potential dynamics. Earlier I mentioned that in this model, the membrane potential is basically just a linear filtering of the spike trains. So that's what I'm showing here. And then this last funny looking term is just encoding the Poisson spike, the conditionally Poisson spiking of the neurons. And so the only reason I'm showing the full form of this action is so that we can contrast it with the central object of this non-perturbative renormalization group that I'm going to get to in a second. Were there any questions before I move on? So two reasons. One is that we could view this as we could just take this to be the model for the membrane potential dynamics. The other is that I could, even in the leaky network, I could add some time dependence here in the JIJ. But basically what this form that I'm showing on the slide here is I've just formally solved for V using an integrating factor. And so the explicit time dependence here is an exponential filtering, but it doesn't need to be an exponential filtering. I could generalize my differential equation to have a temporal JIJ filter here, and I would get basically the same thing.
So I guess I would have thought that there was actually an equation.
So you could put the equation of motion there instead, and that's what you would have to do if we had some nonlinear membrane potential, nonlinear part of the membrane potential dynamics. In this case, we can solve formally for V in terms of the spike trains. So it ends up just being a little bit easier to do that for this analysis. But in general, we would replace that with the differential equation. Yes. It's going to be symmetric in this analysis. I will comment very briefly on a possibility for looking at asymmetric matrices. If we make it asymmetric, there's a step in the calculation that becomes very difficult to do because it relies on being able to not only diagonalize the matrix, but I want to be able to diagonalize the matrix, but the calculation becomes tractable when the transpose of this matrix is equal to its inverse. Because then basically there's a trace that you can get rid of those projection matrices, where for an asymmetric matrix, you can't do it as easily, which makes it much more difficult to take the n goes to infinity limit to write down a tractable equation. But I'll hopefully have time to come back to that point at the end. So for now, we will be restricted to the tractable case of symmetric matrices, but I think there is some promise for going beyond that. But it's mostly a technical obstacle, not a fundamental obstacle, I think. All right. So last point, the other point about this action is that those mean field equations that I mentioned, you can obtain from this action by taking functional derivatives with respect to the different fields. So if you take functional derivatives of this action and demand that they're equal to 0, you would recover those mean field equations that I showed earlier. Now, the central object of this non-perturbative renormalization group that I'm going to be talking about is something slightly different called the average effective action. It's not the average of the action, but it's structurally related to it. And the idea behind this average effective action is that if we wanted to write down the actual equation for the means, so what is called mean field theory, that is actually an equation for the most probable path, or the most probable dynamics of the system, which if it's a Gaussian process, the most probable dynamics would be basically the average dynamics. But if you actually want the average dynamics, you don't get that by maximizing the action. You get it by maximizing this other quantity called the average effective action. And so the saddle points of this will give you the true means. And this is a functional that does encode all information about the statistics and the responses of the network. So this average effective action, if we could calculate that, we would know everything about the network statistics. Now, of course, such a powerful, all-encompassing function means that it's almost impossible to calculate in practice unless you're just dealing with a nice linear Gaussian process. But it turns out to be convenient because it shares a lot of its structure with that action that I showed on the previous slide. And that gives rise to this renormalization group idea, which is what we're going to do is we're going to create a one parameter family of models that interpolates from a limit in which we can actually calculate this average effective action to the true model that we actually want to derive this quantity for. And in particular, the solvable limit of the model is going to be the mean field theory, and that's the limit in which this average effective action would just be equal to the effective action. And then we're introducing this parameter lambda, which is going to allow us to tune from that mean field theory to the true theory. And the way we do that is we're going to derive a very horrible differential equation where basically the mean field theory is the initial condition and the true theory is going to be the solution. And the idea is that we want to carefully regulate the degrees of freedom of this model in sort of a hierarchical fashion. And so Wilson's idea for the renormalization group was we want to calculate these functions that would encode all of the information about the statistics of this process. But doing that calculation all at once, so integrating over all of the degrees of freedom at once is generally impossible. So his idea was, well, what if we integrate over some of the degrees of freedom a little bit at a time? And the idea is that you integrate over some of the degrees of freedom, you figure out a new model and how it relates to the old model. And then you iterate this process until eventually you've integrated over all of the degrees of freedom. So that's the idea behind this method. Now, as I mentioned before, now we have to ask what are the appropriate degrees of freedom for a network model? Because in the case of a lattice, the degrees of freedom are spatial degrees of freedom, or the inverse of that, typically called the momenta. And so this picture that you may see is that you start by integrating out the small length scale degrees of freedom. So you integrate over those degrees of freedom and see how they contribute to this function gamma that you want to calculate. And then you integrate over the next set of length scales and the next set until eventually you've integrated over them all, and you have an estimate of this object that you want to calculate. But as I mentioned, the brain is not organized as a nice crystalline lattice. It has random looking connections, or we often model them as random. And so we don't want to express the degrees of freedom in terms of momenta, but then what is the appropriate basis in which to organize these degrees of freedom? And one possible answer is that it turns out that we can just use the eigenvalues of the synaptic weight matrix. And then in that case, we're going to choose this parameter lambda to be a threshold on the eigenvalue. So we're going to integrate over degrees of freedom up to this eigenvalue threshold lambda. We can do that, becomes simpler in the case of the symmetric Jij, because then we know that the eigenvalues of the synaptic weight matrix are purely real. If we had asymmetric Jij, those eigenvalues would be complex, but you could still take this to be the threshold on, say, the magnitude of the eigenvalues or something like that. Now, again, this is just an answer. Other ideas have been put forward. For example, Bill has a paper with Serena Braden from 2017 where they were using the eigenvalues of the PCA decomposition to do a kind of coarse graining and an icing-like model. And then more recently, Klein and Palmer had a paper using this non-perturbative renormalization group idea applied to Gaussian processes, but interpreting it as sort of in terms of this information bottleneck that some of you may be familiar with. And so they determine degrees of freedom based on this information bottleneck. But what we're going to consider today is just a relatively straightforward thing, because we're going to assume that we know what the Jijs are, so we know we can write down what the distribution is, and so we're going to regulate those eigenvalues. All right. Any questions about that?
May I ask a clarification?
Sure.
So if I understood correctly, I mean, the true final action or the true final result or exponent or whatever depends on the mean field you start from, or is independent? Because there are many mean fields that I can construct as initial guesses.
So it starts from the specific mean field that you get by taking the saddle point of the bare action. So if I take the saddle points of this action, that's going to give me a set of equations. And so this bare action that I'm showing on the screen now, that is going to be the initial condition, and that's what I mean by the mean field theory.
I see. So, but if you change that action, that initial one, I mean, essentially you can change the final true answer.
So in principle, this procedure should be independent of your starting point, because the idea is that we construct this flow of models so that no matter what, we end up at the true theory. Now, in practice, we're going to have to make an approximation or set of approximations which can introduce some artifacts into this procedure. But in principle, the starting point is just chosen to be any convenient starting point, and the mean field theory ends up to be the most convenient in this particular model.
Okay, thanks.
No problem. All right, so this quantity that we would like to calculate, this average effective action, turns out to satisfy a horrible differential equation called the Wetterick equation. And it's horrible because this function gamma is really a functional, not just a regular function. And this is really so this is a nonlinear functional partial integral differential equation. It's nice in that it's exact. So to get to this point, one doesn't have to make any approximations, but exact usually means useless without making some kind of approximation. And so, but the reason this is useful is that because we know that this object gamma shares a lot of its structure with that bare action, that allows us to make something like a variational guess for what it should look like, which is going to allow us to reduce this horrible equation to something a little bit less horrible. but something that we can solve at least numerically. And so the strategy is to use symmetries of the model or derive certain identities and use some physical intuition to come up with a guess for what this functional gamma should look like, plug that into this equation, and reduce it to something that we can actually solve on a laptop. And so we can show that this functional gamma has a general form that's on the screen now for you to contrast with what the bare action looks like. And so if you compare these, you'll see that they look very similar. They both have these response fields. I've changed the notation of the fields in the bare action because these are, you can think of them as the means of the corresponding fields in the bare action. So we still have this set of response fields. We still have these terms that are basically encoding the membrane potential dynamics. And then the last term here, which I said was Poisson spiking in the bare action, is now some functional that we can't restrict it any further. So what we would say is that the Poisson spiking is renormalized by the fluctuations of the model. And so this final functional in gamma is the thing that we have to try to calculate. And by comparing these, that gives us an idea for how we might restrict the form of that. So notice that the Poisson spiking term is a local functional, meaning it only depends on one single neuron index and one single time index. And so that motivates an approximation where we're going to assume that this functional epsilon here is also a local functional of neurons and time. So we're going to say that gamma is approximately all of those unrenormalized terms, plus this final piece, which now we're going to parameterize as a sum over all neurons, integral over all time, but it's a sum and integral of now just a pure function of the spike response field, which is nu tilde here, and the mean membrane potential, which I've labeled psi here. And this is the thing that we're going to plug into that Wetterick equation. We have to, and this is where the n goes to infinity limit comes in, where the more and more neurons comes in. We have to evaluate a bunch of time integrals. We have to evaluate a bunch of matrix traces. And once all of the smoke clears, what we get is a nonlinear partial differential equation for this unknown function u. A couple things about this partial differential equation. This is almost the recursion equation that you would get in a typical renormalization group calculation. I'll clarify on the almost when we get to part 2. But the main things to note, some of the main things to notice about this equation are that it turns out to only depend on the distribution of eigenvalues of the synaptic weight matrix Jij. Turns out that at least at this level of approximation, the eigenvectors fall out of the equation, which is nice because we know what the eigenvectors are for lattices, they're Fourier modes, but for a random neural network, we don't know what those look like necessarily. So it turns out to be super convenient that they fell out of this equation. And it's a nonlinear equation, but it's something that we can try to solve on a computer. We can't write down an analytic solution, but we can solve it numerically. It's a little bit easier to solve it if we make an additional approximation, which is sort of in the spirit of a moment closure approximation. But I'm not going to go into the gory details there. All I want to point out about the solution of this equation is that effective nonlinearity that I mentioned that we want to calculate, we obtain from a derivative of this function. So we take the derivative with respect to the first variable, which corresponds to the spike response field, set that to 0. That is our estimate of this effective nonlinearity that we want to calculate. Yes, thank you. So the superscripts here correspond to derivatives with respect to the two variables x and y. So first index corresponds to x, second index corresponds to y. So it's.
I guess I would have expected that flow equations depend on
We'll get there. That's the almost. So again, this is not, for those of you who know about the renormalization group, this is not the equation where we expect to get fixed points, because so far I've only done, there's two steps to the renormalization group procedure. The first one is coarse graining, or the regulation of the degrees of freedom. The second one is rescaling. I have not done the rescaling step yet. That will come. So I'm just going to show you some examples where I've solved this equation for a couple of different networks. First, excitatory lattice network, so a three-dimensional torus, and then we'll look at a random network. So first, I'm going to look where the parameters are tuned to be in the subcritical regime. And so what I'm showing here, so on the left is the eigenvalue density for a 3D hypercubic lattice. And then this plot here is the effective nonlinearity calculated with this non-perturbative renormalization group method in red. The blue data points that you can see kind of behind that are from simulations of this of the actual stochastic network model of 1,000 neurons. And then the black dashed line is what you would expect to get from mean field theory. And so what's being plotted here is essentially so the data is a scatter plot of the mean firing rates of the neurons in the model against their mean membrane potential, because according to the analysis that I went through earlier, that scatter plot should map out this effective nonlinearity. And so we're looking at, again, a subcritical regime where you get a slight deviation from the mean field prediction. So qualitatively, not a whole lot changed, but you can see that the gain of the effective nonlinearity has been reduced slightly compared to the so-called bare nonlinearity. And we can look at the same thing for the Gaussian random network. Yes?
Why is that the density is not symmetric in the hypercubic three-dimensional lattice, or is just my eye?
It's not symmetric because for the 3D lattices, I added that soft reset term. So that reset is the same for all the neurons. And so that's just a constant shift of the eigenvalue density.
Okay, thanks.
And then I could do that could have done that for the Gaussian random network as well, but I didn't for simplicity. But we can calculate the this effective nonlinearity for Gaussian random networks. So this is a fully connected network, although you could add some sparsity as well, where each synapse is just chosen from a Gaussian distribution with zero mean and variance equal to half of this quantity lambda max, the maximum value of the eigenvalue here. So this is what it would look like for a subcritical network. And again, it's not that far off from the bare nonlinearity, but we see that the gain is reduced slightly. And so in the subcritical case, well, what happens is that, so I'm parameterizing these cases by the gain of the bare nonlinearity, that's lowercase derivative of lowercase phi at v equals 0, times the maximum eigenvalue of the synaptic weight matrix. And when that's less than some critical value, we're in the subcritical regime, and the gain of the effective nonlinearity is less than 1 over this maximum eigenvalue. Now, when we go to the critical case, and so these simulations are close to the critical case, maybe not exactly tuned to it, but the idea is that there will be some critical value of the bare nonlinearity and synaptic weight matrix at which the gain of the effective nonlinearity turns out to be 1 over the maximum eigenvalue of the synaptic weight matrix. And so we're starting to see there's more deviation from the bare nonlinearity or the mean field theory. The non-perturbative method is agreeing pretty well with the simulation data. And then if we increase the strength of the synaptic connections further, we're going to get into the supercritical regime. And so in the supercritical regime, something interesting happens. One is, so this is when the bare value of the gain times the synaptic lambda max is greater than this critical value. But if we look at the solution in red from the non-perturbative method, what we find is that the gain of the effective nonlinearity actually gets pinned at 1 over lambda max. So it doesn't keep growing. And that will be relevant. I'll explain the relevance of that momentarily. Now, you're probably noticing that the prediction of the method does not match up with the simulated data very well here. That's because in the supercritical regime, the reason that the slope is getting pinned at this 1 over lambda max value is actually there is an extended region here that is perfectly linear. So this nonlinearity has become non-analytic once we get into the supercritical regime. And it's very difficult to numerically integrate a partial differential equation through a regime where a non-analyticity develops. So we had to sort of weaken the approximation to get the numerical results here, but this still gives us a qualitative picture of what's going on in this supercritical regime. And this is basically this regime in which the effective nonlinearity has become non-analytic is the regime in which we can get phase coexistence in the network. And the reason for that is that now if we look at the effective nonlinearity here, where we're in this supercritical regime, and we have this extended linear region of the network, well, now if we try to find the steady states of this network, or at least of the mean dynamics, now instead of just having 2 points where the V over J curve intersects the nonlinearity, we have a whole segment of this line. And so there are 2 extremes of that line, which would correspond to extremal metastable states, but then there's this whole line in between, which represents phase coexistence. And those of you familiar with the Ising model may recognize that this looks a lot like what you get if you look at the free energy, where there's this, in the supercritical regime, you have this flat plateau in the free energy, which corresponds to phase coexistence. And so that's the same thing happening here in the spiking network model, that in the supercritical regime of the spontaneous networks, you get this regime where phase coexistence is now possible. And what that means is that we could, if we simulate the network, say on a 2D lattice is what I'm about to show you. So I'm going to start the simulation in the subcritical regime, and then at t equals 3, I'm going to increase the gain of the bare nonlinearity, and what we should expect to see is it will form clusters of high activity states and low activity states. And that's indeed what we're going to see in the actual simulations. I think you have to do a little bit more careful tuning to try to get everything to be in that phase coexistence regime, but the idea is that we may be able to achieve a certain phenomena called spinodal decomposition here. And that comes from the possibility of this phase coexistence. Now, this is going to be harder to see in the Gaussian random network, because now we don't have that spatial organization, but you would still be able to see a change from that sort of baseline asynchronous activity to now some neurons being suppressed and some neurons being more active once that increase in gain happens. And so this is coming from now having that phase coexistence region and those 2 different extremal metastable states, although it's much more complicated in the case of the Gaussian random network, because one of the things I want to stress with this result is that we're able to calculate this effective nonlinearity. It doesn't matter whether we're looking at a lattice or a Gaussian random network, but if we want to describe the actual collective activity that's going on, we still have to solve this equation for the means. And this is where now the modes of the eigenvectors of the Jij become important, and also the signs of the Jijs become important. Because if we want to actually understand the dynamics of the full network, in particular the population statistics, then we still have some work to do beyond calculating this effective nonlinearity. In homogeneous excitatory networks, we basically get these extreme metastable states and phase coexistence. But if we have random networks, then if we want to understand the population activity, we probably need to do something like a dynamic mean field calculation with this effective nonlinearity, because again, we have symmetric synaptic weight matrices here, so we'd probably expect some kind of spin glass dynamics in this regime. So we've calculated this effective nonlinearity, but we would still need to either simulate this equation or do some other kind of calculation to say something more about like the spin glass behavior. All right. Any questions about this first part before we get to the universality bit? Yes. So in the theoretical analysis and the mathematical analysis, a phase transition occurs at this point where the non-analytic behavior develops. So in the subcritical regime, that effective nonlinearity that I was calculating, perfectly analytic. But when you get to the critical point, that's where you get the development of a non-analytic behavior in the solution. And then you go into the supercritical regime and that non-analyticity just grows larger. And this will come back in the next bit of the analysis, because that's how a phase transition is, defined in physics, is that it's the thermodynamic limit, and then you have a non-analyticity develops at a critical point. So it's for infinite systems. For finite systems, strictly speaking, you don't have a true phase transition, but you can then view the size of the system as another tuning parameter. And you can ask, well, if n is large enough, we would see signatures of these sorts of phase transitions. And you can ask how large is large enough. And that's a very good question to ask, because we know there are certain types of phenomena that seem to require finite n. So for example, jumping between different metastable states becomes very hard in the infinite size limit, because the barriers between those different states basically become infinite. But one can tune away from the infinite limit to try to understand what's going on in more realistic finite systems. In what I'm showing you today, no, because that goes back to this restriction to symmetric matrices, because I can't make a symmetric matrix that obeys Dale's law. But as I'll mention on one of the future work slides, that's an obvious case that we would love to be able to do this sort of calculation for. But right now we're still building the foundation so that we can actually get to that case. All right. Anything else?
So can I ask for a clarification again? I mean,
Sure.
we're talking about phase coexistence. I mean, are you implying that there is a first order transition of some kind with nonlinearity, or is something else? Because you're talking about, criticality, but now you're talking about phase coexistence. I mean, what is really, this point?
So I think I will answer that question in the next part.
Okay.
So I'll ask for your patience on that point.
Thanks.
All right. So again, this analysis so far has, we've calculated this effective nonlinearity using this method, but as I mentioned, this was only the first part of the renormalization group procedure. If we want to talk about universality and fixed points, we need to do the second step, which is rescaling, which is another thing which I think has been a barrier in figuring out how to apply these renormalization group ideas to spiking networks, because normally if you get taught renormalization in the context of a statistical physics class, where you're thinking about, say, just to make it easier to draw, the 1D icing model, this regulation of the degrees of freedom, so they integrate out every other spin And then the next step is to rescale the system so that you put the lattice size back to what the original lattice was. So you'd have, for example, some other neurons over here, and then you rescale all of the lengths so that the system looks kind of like what the original system that you started with is. But how do you do that for a network? A line or a square, you can do this procedure for a network. It's not clear what the rescaling step you need to do is. And at least in terms of this flow equation that I derived, the way you figure out what the rescaling is we're going to make a change of variables to render this flow equation approximately autonomous, at least as we're getting close to lambda goes to lambda max. And so we need to make a change of variables. I'm going to pull out separate the part that corresponds to the critical gain of the nonlinearity. And so I'm going to have this leftover piece that I'm going to call the dimensionless potential. And it's the dimensionless potential that I'm going to derive a related flow equation for, that is the one that has fixed points, that we can do things like look at the linear stability and estimate these critical exponents. So this is where the logarithm that Bill asked about comes into play, because I'm going to index this dimensionless potential by something conventionally called the RG time, which is the logarithm of the difference of eigenvalues, basically. And then I've also introduced a number of quantities, omega, x, and y, which are running scales. So these are, so I'm rescaling the variables, the spike response field and the membrane potential variable y here by some quote unquote constants, but these constants are going to change with this eigenvalue threshold as I'm regulating the degrees of freedom. And we need to figure out how those should change with the scale. And it's going to turn out, so this is how we're going to be able to define what the notion of dimension is for a network. Because it turns out that if we look at the eigenvalue density of, again, we're looking at symmetric matrices right now, but for the most part, whether we're looking at a lattice or a random network, this eigenvalue distribution has a particular scaling near its maximal eigenvalue. And so if we calculate the eigenvalue distribution for a d-dimensional lattice, you can show that near the maximum eigenvalue, it scales as the difference between the maximum eigenvalue and the threshold to the power of the spatial dimension over 2 minus 1. And so this motivates defining the dimension for an arbitrary network in terms of the scaling of its eigenvalue distribution near that maximum eigenvalue. And so this is going to be our choice of dimension for an arbitrary network. So it's not necessarily the spatial dimension in which the network's embedded, not necessarily one of these other fractal dimensions, but it's this, maybe what you would call the spectral dimension coming from this scaling. And from this scaling, we can work out that coefficient omega needs to scale like lambda max minus lambda to the d over 2 plus 1. And then the product of x and y has to scale like that difference to the d over 2. And so this is the answer to one of the questions I mentioned earlier, which is what is the appropriate definition of dimension for a network? And we can go and plug these scalings into this equation, and we're actually, because on the previous slide, I only pinned down the product of x and y. That allows us to introduce an additional exponent that I'm going to call eta here, which is something that we have to determine as part of the solution, because that's going to be different for the transient networks versus the spontaneous networks. And so this is basically this is like the generalization of momentum shell coarse graining, for those of you who know what that term means, to a more arbitrary network model. And so we plug this into that differential equation for omega, or not omega, u that I showed earlier. And what that's going to give now is a differential equation for this dimensionless potential w that I've defined. And so there's two parts. The right-hand side is just the dynamics of the flow that comes from the structure of the model. It's just a rescaled form of the right-hand side of the equation for U. But now the left-hand side has some additional terms, and this is what's called the dimensional flow. So these are the terms where that allow us to find fixed points for this dimensionless function W that we've introduced. And fixed points meaning if I set this derivative with respect to the RG time equal to 0, then I have a partial differential equation that in principle I can solve, and that solution is going to correspond to a scale invariant critical point. And then again, we're going to have to determine also what this exponent eta is as part of the solution. Now, this equation is too difficult to solve analytically, and it's also numerically very dicey, because if it tends to explode in certain directions. And so the typical way we would solve it, or one of the typical ways, is that we're going to make a truncation. So we're going to expand the dimensionless potential as a series in its two variables, where the coefficients are what I'm going to refer to as couplings. And for the purposes of, not running too far over time, I'm just going to consider what happens keeping the first couple of couplings in this expansion. So basically, we're going to plug this in up, keeping only a finite number of terms, and that's going to give us a qualitative picture of the renormalization group flow of this model. And the last bit is, in order to determine what this exponent eta is going to be, that's where we need to make some choices that correspond to the transient network versus the spontaneous network. So for the transient network, in the interest of time, I'm not going to explain where this comes from, but you impose something called rapidity symmetry on two lower order couplings, which means that we're going to fix G12 equals minus G21 for all time points S. But then in the spontaneous network, we're going to fix a coefficient corresponding to the just baseline noise fluctuations. We're going to fix that to be 1, meaning that that does not rescale as we do this rescaling procedure. So again, for those of you who've done renormalization in something like the 5-4 theory, that would correspond to having the Gaussian part of the action not rescale under your RG transformation. So we do that, we plug this into these flow equations, and what I'm going to show you is some phase portraits for a couple of the different coefficients in the case of the transient network and the spontaneous network. So I'm just going to look at two couplings, G11 and G12. And so what we find for the transiently active networks is, if we look at the phase portrait of these two couplings in dimensions greater than 4, we get something that typically looks like this. So what this is, so each of these lines is showing the evolution of G11 and G12 in this RG time. So for example, if you choose the initial conditions to be where my cursor is, then as you evolve the system in time, it would follow this particular line. And the thing to notice in this phase portrait is that there's a particular point labeled in red here, which is the fixed point. So it turns out to be G11 equals G12 equals 0. And so this is a point where if we initialize the system at that point, it stays there. And going through that point are two important lines. One of them, this sort of vertical line, is called the critical manifold. Because if you look at the line on the horizontal axis, you'll see that all the trajectories close to that flow away from the fixed point, but they're flowing towards the fixed point along this critical manifold. So what that means is that if you tune the properties of the network model, which in this case would be either tuning the gain of the nonlinearity or tuning the synaptic strength, you can tune the model to put it in terms of these rescaled variables right on this critical manifold. And then what happens is that the flow of these couplings would go right to this fixed point. So, but if you if you don't tune the network to be right on this critical manifold, it will flow towards that critical point and then be repelled away from it. And so this is the typical picture of a critical point in one of these RG flow diagrams, is that you have a fixed point with an unstable direction and one stable direction. And those universal exponents are determined by what this fixed point is, but also by the repelling of the flow away from the fixed point. And so in this case, in dimensions greater than 4 for the transiently active networks, your fixed point is trivial. And this is what would give you mean field values of these critical exponents. So for example, for the transiently active network, there was that decay to the quiescent state that went as 1 over the time. But if we look in dimensions less than 4, this portrait changes. And so now that fixed point at 0 has become unstable. So both directions away from that fixed point are unstable now. So even if you start just slightly off of that fixed point, the system's gonna flow away from that. And instead we have these 2 other critical points that have become introduced. And so these are non-trivial critical points, so that if you tune the system to be on this critical manifold, it would flow, the RG flow would be towards one of these critical points, and that's where you get this scale invariant behavior that has these bizarre values for things like the critical exponents that are not predicted by mean field theory. And then again, the flow, if you're not quite at the critical point or on the critical manifold, the flow away from the critical point is going to determine some other sets of exponents that I'll show you momentarily. And so in particular, the flow near the critical point gives something called the correlation length exponent. That's what it's conventionally called because in a lattice system, it would tell you how correlations between the neurons decay with space. I don't know what it would mean in a random network. But I'm going to keep the name correlation length exponent. And so we can estimate that keeping more terms than I showed on the phase plane analysis on the previous slide. And so we can estimate this critical exponent as a function of dimension. And so what I'm showing here are estimates for different orders of truncation. So keeping more and more terms compared to also a perturbative expansion that I'm showing in the dashed line. And so what we get is a non-trivial value of this exponent, and we can estimate it at least down to a dimension of 2.78, below which something funny happens in the network model. I'm not sure yet if if this fixed point becomes unstable to yet another critical point, or if this approximation has just reached its limit of applicability and we need to make a more refined approximation in order to go to even lower dimensions. And then we can also calculate that decay exponent beta that I introduced earlier. And it turns out that at these critical points, that exponent is just the dimension divided by 4 whenever the dimension is less than 4. And we can verify that in lattice models of d equals 2 and d equals 3, where if we tune close to criticality and we look at the decay of the mean membrane potentials with time, that decay, there's a power law region that pretty much agrees with this d over 4 exponent. All right, so I'm going to try to not go too far over time here. So I'll ask that if there are any questions, we hold them till the end for anyone interested. Last thing I'll say about this particular case is we can try to calculate what the avalanche exponents are that I introduced way at the beginning of the presentation. So there's a number of exponent relations that relate the size exponent, that duration exponent, to these 2 exponents I showed you on the previous slide. So we can calculate those as a function of dimension. It turns out that there's no dimension that's very consistent with all 3 of those measures. So dimensions somewhere between 3 or 4 look like they could agree with the size exponent from the avalanche data or the duration exponent, but there was this other exponent 1 over sigma nu z that doesn't seem like it would be predicted by this spiking network model. And so that might suggest that the avalanche dynamics seen in experiments are not consistent with this this particular universality class, but there's a number of caveats here. Again, this analysis was done for symmetric connections. The real sliced tissue is probably not symmetric. Questions about what would happen in lower dimensions. And then, as I mentioned much earlier, the linear membrane dynamics do have an impact on one of the critical exponents. And so if we include nonlinear membrane dynamics, which we would expect to be there in the actual model, that may change some of these estimates as well. So that's a next step to improve this calculation. But the fact that we can calculate these exponents in the first place and non-trivial values of them, I think is an important step towards that. All right, and now, again, we're at the top of the hour, so I will try to very briefly mention what happens in the case of spontaneous networks, because this is where we get a surprise that's related to one of the questions from Zoom a few minutes ago. So we can look at a similar flow of the couplings for the spontaneous network model, and we get something a little bit strange. In dimensions greater than 6, we get that a trivial critical point at 0, 0, and we get a couple of other fixed points that are completely unstable. So they wouldn't determine criticality or critical properties of the network. But then when we go to dimensions d less than 6, this fixed point at 0 is no longer stable, but these other fixed points have disappeared because they collided with the fixed point at 0, and now we have a phase portrait that doesn't seem to have any fixed points at all, other than this trivial fixed point that's not a critical point. So it turns out that there's no fixed point for real valued G11 and G12. But if you assume that G12 is imaginary, then the phase portraits look very different. And when D is greater than 6, now there's just that stable fixed point at 0, 0. And in dimensions less than 6, now we get 2 new critical points. But they're critical points at imaginary values of this coupling G12, which, when that happens, so you have a complex valued fixed point. You might wonder, is this even meaningful? Is this just a non-physical solution of this analysis? And there's an argument to be made that this is actually a physically meaningful solution, because we've seen this before in other models. And we can also show that the critical exponents that we would actually measure are real. So even though the model has this complex valued fixed point, the things that we would actually measure turn out to be real. And the example in which we've seen similar complex valued fixed points before is at something called a spinodal point, which is a type of first order transition, which would come up, for example, in something like the icing model, but where you've explicitly broken the symmetry. Another model that some of you may be familiar with is called the phi-3 theory, or the Yang-Lee theory, which is thought to describe this sort of first order transition. And this is different from what people normally expect when talking about phase transitions, is that typically they're thinking of a continuous or so-called second order transition, which is where you get all these nice universality properties. But it turns out that this sort of first order discontinuous transition, even though it's first order, it still has these quantities that are universal. And there's a series of papers by this author Zhong, who does various renormalization group analyses for this phi-3 theory to show that you get these complex valued critical points, but they describe what the transitions that you would observe in simulations. And so this is one of the surprises that we might have thought that the spontaneous network would be something icing model like, and while it kind of is, it's icing like in the sense of the symmetry broken icing model where you have a first order transition and not this famous second order transition that I showed at the beginning. And with this method, we can estimate things like this running exponent eta as a function of dimension, which agrees well with another non-perturbative renormalization group method of this Yang-Lee phi-3 theory. A note about this exponent is that it's determined entirely by the critical point itself. This other exponent, the correlation length exponent, is determined by the repulsion of the flows away from that critical point. And it turns out that that exponent is much more sensitive to some of the details of this local approximation that I made. And in particular, we get an estimate for this correlation length exponent that is non-monotonic, which is probably not realistic. And in this other study of this spinodal transition, what they found is that you actually need a more complicated non-local ansatz guess for that function in order to capture what's going on in much lower dimensions. So that suggests that, you know, we need to make a more refined approximation to quantitatively estimate these exponents in lower dimensions, especially, you know, 2 or 3, which may be closer to the physical dimensions we might expect to see. But that it at least tells us that we might expect the spontaneous network may not have a second order transition, but it has this kind of first order spinodal transition. And let me skip this to just get to the summary. So we've extended this non-perturbative renormalization group method to apply to a kind of leaky integrate and fire model of spiking dynamics. We can calculate certain quantities like this effective nonlinearity, and we can calculate some properties of the universal features like these critical exponents and say something at least preliminary about the different universality classes of transient versus spontaneously active network models. I wanted to end just by mentioning a couple possible, whether this has possible implications for this critical brain hypothesis. So one is that there's potentially these different universality classes in vitro versus in vivo. Another is that, we might wonder, can we try to estimate this effective dimension from inferred connections between neurons? So there's lots of methods for inferring not the true connections per se, but some notion of functional connections, like we heard in the first talk today. And we might ask, well, if we look at the spectra of eigenvalue spectra of those connections, could we estimate something like this effective dimension? Now, there's definite confounds there. We don't know if the inferred connections, how they're related to the ground truth connections, but it gives us something to potentially look at. And then I also want to say that this analysis has nothing to say about this idea that the brain is actively regulated to be near a critical point. So the synaptic connections in this model are fixed in time. One could imagine if you added synaptic plasticity dynamics to the model so that these JIJs also evolve in time, well, that's also going to do something to these renormalization group flows and the behavior of the model. And who knows what happens there? Maybe some of these critical points actually become fully attracting, and that would be, that might correspond to something that is sometimes called self-organized criticality, where you would have a critical point, but that one that is actually fully attracting in the renormalization group flow. But this method does not have anything to say about that. That is hopefully something that could be analyzed at some point in the future. So I'm over time now, so I have some things to say about the next steps to get this closer to more realistic networks, including those nonlinearities, looking at asymmetric networks, and treating the effects of the synaptic weight distributions at this RG level instead of at the level of the effective nonlinearity, but we're over time. So perhaps people who want to mill around or are coming to dinner can ask about those later. So I'll just end here by thanking you all for your attention, thanking funding from Stony Brook University and the NIH, thanking my lab for enduring practice runs of this talk. And then if you have follow-up questions later, here's my email on the screen. Here's the archive identifier for the preprint that has all the gory details in it. And thank you again for your attention and sticking around till late on a Friday afternoon to hear about some hardcore neuroscience theory.
Can I ask some questions?
So someone in the live audience was going to ask first, and then we'll get back to you.
Okay. So the question was, is there any way to try to estimate the effective dimensionality without trying to infer the neurons or infer the connections between neurons? And the answer is a definite maybe. And I think this also relates to one of the questions that someone asked you about criticality in your talk. So in this particular model, we can actually, we can relate the spike train covariance to the eigenvalues of the Jij matrix. And so in particular, the correlated, or the time integrated correlation. So if I integrate the correlations, the cross covariances of the spike trains over time, that gives me some set of correlation coefficients. I can look at the eigenvalues of those correlation coefficients, and those eigenvalues are going to be proportional to, well, in a certain case, they will be proportional to the eigenvalues of or 1 over 1 minus the eigenvalues of the Jij matrix, times the gain of the effective nonlinearity squared. At least in the case where, so let me just consider this for the case where I've somehow tuned the network to be so that all of the neurons are in this sort of supercritical regime where they have zero, their expected membrane potential is 0. So in that case, the gain of the nonlinearity is 1 over the maximum eigenvalue. And so if we know the distribution, so at least theoretically, if we know what the distribution of the synaptic weight matrices is, we could work out what the distribution of these eigenvalues is. And in this case, where the maximum of the synaptic weight matrix is lambda max, the maximum of the correlation eigenvalue is going to be infinite. And you can work out how this decays. And it goes something like, it falls off as the power of the eigenvalue to minus d plus 6 over 4. So potentially, if you measure the eigenvalues of the correlation matrix, if it were in this particular supercritical regime or in this phase coexistence regime, then maybe you could infer the dimension that way. And then over lunch, I was thinking about the question about criticality that was asked to Carsen. And you can correct me if I'm wrong, but the plot where this is the eigenvalue rank versus the eigenvalue, is that correct? So if you work out
what that is for the scaling, then I believe what you get is that the correlation eigenvalue would go like the rank 2 minus 4 over the dimension plus 2, which if d equals 3 is 0.8, and if d equals 2 would be 1. So whether that is actually what could be giving rise to this power law decay of the eigenvalue distribution in a spontaneous cortex, I don't know, because there's some assumptions that I'm making here that you've gotten the network into this sort of phase coexistence regime and that their membrane potentials are mostly confined to near v equals 0. But so potentially you could estimate some notion of dimension this way, and maybe it could be an explanation for that rank decay.
If I remember correctly, the mapping is that the behavior of the density is the distance to the maximum to the d over 2 minus 1.
Yes.
The d equals 2 is very weird. That corresponds to it coming in flat.
Yes, but basically it's
Finite density.
yes, exactly. So, and it's known that in many systems d equals 2 gives strange critical behavior. So many weird things could happen near d equals 2, and so this is just a
and just the ordinary.
Yes, exactly. So because so Figner semicircle distribution is square root, that would be effectively d equals 3 for a Gaussian random matrix, a symmetric Gaussian random matrix. All right, so there was a question on Zoom.
Yeah, can you hear?
Yes.
Yes, so the exponents that you were presenting, they were all in regular lattices, in regular networks, or did you try to see those exponents in random networks?
So they're going to be most easy to characterize with simulations in the lattices, or perhaps something like the maybe the random regular network. To do the full renormalization group analysis in the random network case, there are some additional steps that you would need to take that I've not talked about today. One way to do it at the level of this non-perturbative renormalization group flow is that you would actually also have to calculate the flow of something called disorder cumulants. And this has been done for something called the random field Ising model. And so in principle, that could modify what you would see when you actually try to measure these exponents in experiments. And so that is a much harder calculation. So I've not tried to do it yet, but it's known that what's called in the physics literature disorder, that can be at times what's called a relevant perturbation to the renormalization group flow. And so that could give some additional contributions to the exponents that I don't think would be reflected here. Another way to try to understand the effect would be to do something like I mentioned earlier, like a dynamic mean field analysis of the equation for the true means that's shown on the slide here, where you're basically transforming heterogeneity in the population to an effective time-dependent noise. And you could potentially study, like this has been done at the sort of level where instead of using the effective nonlinearity, you just use the bare nonlinearity. Unknown what happens when you get the nonlinearity that actually depends on what the strength of the synaptic connections is, and when it develops this non-analytic regime, or how that singular piece of the firing rate function affects that when you're at the critical point. So more work to do there for sure. And
Yeah.
hopefully it won't be too intractable.
Okay, so thanks. So I have another question. Now that you have this equation here, do you try to modify this equation and add some external potential or something like that?
It depends what you mean by external potential. Like we can adjust the value of the, like if you're imagining, say, an external tonic current or slowly time varying current, that could be included in the sort of rest potential effectively. And you could study things like hysteresis in the model. So if you provide a driving current to the model, you can observe hysteresis effects, at least in the excitatory lattice and the random regular graph. But if you mean membrane nonlinearities, then I think the similar approaches would work. You lose some guarantees on this approximation that you make, but it should in principle also work if we introduce nonlinearities into the membrane potential dynamics.
Okay, but I mean, your renormalization group analysis, I mean, your flow diagram, I mean, do you have any idea how it changes?
In the case where we add the membrane potential nonlinearities, the only qualitative statement I can make is that there's another exponent conventionally called the dynamic exponent, labeled by Z or Z. And at the mean field level, that would be 2. Typically, at the non-mean field level, it takes on some other non-trivial value. But due to the linear dynamics in the analysis I presented today, it turns out that is always 2, even at these non-trivial critical points. And so I would expect that particular exponent to at least change if we add something like the membrane potential nonlinearities. But beyond that, the other thing I can say is that there's a paper cited in the last bullet point here, which is a recent PRL by Tiberi et al., where they're considering a firing rate network model driven by just some Gaussian noise. But you could view that model as sort of similar to the model that I presented today, where you've marginalized out the spiking dynamics. So you would just have a model for the membrane potentials. And they effectively include a membrane potential nonlinearity, and they show that if that nonlinearity is tuned in such a way that it balances some of the contributions from the firing rate nonlinearity, they can show you also get non-trivial critical points in dimensions of d less than 2. And so I think if one included that membrane potential nonlinearity, that sort of fixed point would also show up in this model. But beyond that, I don't have any qualitative expectations, at least not that I can remember, for what happens if you add some membrane nonlinearities.
Okay, thank you.
Thanks for your questions. All right. Thank you.